Hello students, in this lecture we will learn about chromosomes and their structure. Let us start with the introduction. The genomes of most eukaryotes are much more complex than the genomes of prokaryotes, and the organization of DNA in genomes of eukaryotic cells is also different from the genome organization in prokaryotic cells. The genomes of prokaryotes generally consist of a single chromosome made up of a circular DNA molecule. In contrast, the genome of eukaryotes are composed of chromosomes in the nucleus and in the organelles, that is, mitochondria and chloroplast. The organellar chromosomes are made up of circular DNA molecules, while the nuclear genome contains multiple chromosomes, each consisting of a linear DNA molecule. These DNA molecules are long molecules and are packaged in a unique way to fit inside the small nucleus. For example, the total extended length of DNA in a human cell, which is about 2 meters long, is packaged by tightly binding to small basic proteins, that is, histones, to fit inside the nucleus with a diameter of about 5 to 10 micrometer only. Similarly, the large circular DNA molecules of prokaryotes are also packaged to fit inside the small cell. Let us now see the structure of bacterial chromosomes. Most bacterial cells contain one large circular chromosome. A typical E. coli chromosome has circumference of about 1.6 millimeter, and this DNA molecule is present inside the bacterial cells, which are only about 2 micrometer long and about 0.5 to 1 micrometer wide. To fit into the tiny space inside the cell, the DNA molecule is packaged with DNA binding proteins to form the bacterial chromosome. This bacterial chromosome has no similarity with the eukaryotic chromosome but represents a compact structure like them. The circular chromosomes of bacteria are also called nucleoid. The free DNA molecule occupies a large volume due to the charge repulsion between the negatively charged phosphate groups. These repulsive charges of the phosphate groups are counteracted by the association of the DNA with positively charged polyamines such as spermine and spermidine inside the cell. The circular DNA molecule of E. coli genome is supercoiled, which can occur when additional turns are introduced into the DNA double helix, in which case called the positive supercoiling, or if turns are removed, called negative supercoiling. The supercoiling of E. coli chromosome is generated and controlled by two enzymes, namely DNA gyrase, which catalyzes the ATP-dependent supercoiling of DNA, and DNA topoisomerase I, which removes negative supercoils. Along with the DNA gyrase and DNA topoisomerase I, a set of other DNA binding proteins are believed to constitute the protein component of the nucleoid and play a role in packaging the bacterial DNA. The bacterial DNA binding proteins are usually histone-like basic proteins with about 90 residues. Examples include the HU protein in E. coli, integration host factor or IHF of Enterobacteria, histone-like nucleoid structuring proteins, etc. These bacterial DNA binding proteins not only stabilize the bacterial DNA but also play many important roles, including the regulation of gene expression. For example, the HNS HU protein is a small histone-like protein of about 10 kilodalton and about 60,000 HU proteins are associated with each E. coli DNA. It acts like eukaryotic histone proteins by forming a tetramer around which approximately 60 base pair of DNA becomes one. HU protein has an alpha helical hydrophobic core and two positively charged beta ribbon arms. The arms bind to the minor groups of double-stranded DNA with low affinity, while a component of the alpha helical core interacts with altered DNA such as junctions, nicks, gaps, forks, and overhangs with high affinity. HU protein has been implicated in DNA replication, recombination, and repair, and also in transcription and translation in bacterial cells. HNS is a protein with molecular weight of about 15.6 kilodalton that binds to DNA with an intrinsic curvature, and it helps in the regulation of bacterial transcription in bacteria by repressing and activating certain genes. Integration host factor or IHF is another nucleotide associated protein found in gram-negative bacteria. It is a heterodimer with molecular weight of about 20 kilodalton and composed of alpha and beta subunits only, and its binding bends the DNA approximately by 160 degrees. Most circular bacterial chromosomes has a single origin of replication or the location at which DNA duplication begins. Next, let us take up the structure of eukaryotic nuclear chromosomes. Although the number of chromosomes and ploidy level may vary in different eukaryotes, the chromosomes in eukaryotes have a basic similar organization. The DNA present in the nucleus of eukaryotes is divided between a set of different chromosomes. Each chromosome consists of a single linear DNA molecule associated with proteins that is packaged to form a compact structure called chromatin in the interphase nucleus. The chromatin organizes to form the highly condensed chromosome during mitosis. Hence, the structure of eukaryotic chromosomes is dynamic and it undergoes condensation and decondensation at different phases of the cell cycle and also at different regions of the interphase chromosomes during gene expression, DNA repair, and replication. The packaging of nuclear DNA into chromosomes is accomplished by the association of DNA with DNA binding proteins called histones. Chromatin is the complex formed by the association of DNA with histones and other non-histone chromosomal proteins. Histones are a family of basic proteins rich in positively charged basic amino acids that interact with the negatively charged phosphate groups of DNA. 5 major types of histone proteins, namely H1, H2A, H2B, H3 and H4 are present in association with eukaryotic DNA in all eukaryotic nuclei. The chromatin of interphase nucleus resembles beads on a string, where the string is formed by a thin linker DNA connecting the bead-like structures called nucleosomes. Let us take up nucleosomes. Nucleosomes are about 11 nanometer in diameter and they form the primary structural units of chromatin. Each nucleosome contains 8 histone protein molecules with 2 each of histone 2A, 2B, histone 3 and histone 4. These 8 histone proteins form a barrel-shaped co-octamer around which 140 to 150 base pair of DNA wound twice to form the nucleosome particle. The core histone proteins of the nucleosome contain flexible amino terminal of 20 to 40 residues that extend from the globular domains of the protein. These N-terminals contain many positively charged lysine groups which may interact with the negatively charged phosphate groups in the DNA of the same nucleosome or with those of linker DNA and other neighboring nucleosomes. These N-terminal lysines undergo enzyme-catalyzed reversible acetylation and deacetylation at specific lysine residues. As a result, the positive charge of the acetylated lysine is neutralized and it cannot interact with the phosphate group of the DNA, inhibiting further condensation of the chromatin. The next feature is the linker DNA. Each nucleosome is separated by a linker DNA of 50 to 70 base pairs. It is associated with another type of closely related histones which are collectively called linker histones. For example, H1A to E, H1 degree, H1T and H5 of vertebrates. A single linker histone is attached to each nucleosome to form the chromatosome and it acts as a clamp holding the coiled DNA and nucleosome together. The next level of structure is the 30 nanometer fiber. The chain of nucleosomes joined by linker DNA resembling beads on a string represents an unpacked form of chromatin found infrequently in living nuclei. Generally, the nucleosomes associate to form the 30 nanometer fiber which is called so as it is approximately 30 nanometer in diameter. The 30 nanometer fiber is the major type of chromatin observed in the interphase nucleus. The formation of this 30 nanometer fiber is described by two models, that is the solenoid model and the zigzag model. According to the solenoid model, the 11 nanometer fiber of string of nucleosomes is arranged like a solenoid with the linker DNA arranged in the center without passing through the central axis. However, in the zigzag model, the linker DNA passes through the central axis of the fiber and forms a zigzag pattern of arrangement of the nucleosomes. The 30 nanometer fiber is stabilized by the binding of the amino terminal of histone tails with adjacent histones in the 30 nanometer fiber. For example, the negatively charged histone fold domains of histone H2A interact with the positively charged amino terminus tail of histone H4 of adjacent nucleosomes. Next is the chromatid. Among the non-histone proteins involved in organizing the structure of chromosomes, the chromosome scaffold are a group that plays major role in further condensation of the 30 nanometer chromatin fiber. Long loops of 30 nanometer chromatin fiber are anchored to the flexible chromosome scaffold to form the structure of the extended interphase chromatin. Further coiling of the scaffold into a helix and packing of the resulting helical structures produces the highly condensed structure of the chromatid with a diameter of 700 nanometer. The next structure is the metaphase chromosome. The metaphase chromosomes are formed after the replication of DNA in the cell cycle and therefore contains two chromatids, each representing a chromosomal DNA molecule. The two sister chromatids joined at the centromere forms the structure of the metaphase chromosome, which is about 1400 nanometer wide. Each chromosomal DNA molecule has a centromere, two telomeres and replication origins. The surface of centromere contains a pair of plate-like kinetochores which act as attachment points for the microtubules that radiate from the spindle pole and helps in drawing the divided chromosomes towards the pole during cell division. Centromeres generally contain repeated DNA sequences called alphoid DNA in humans and specialized proteins, for example, CENPA in humans. Another important feature of the chromosome is the telomere or the terminal region of the chromosome. Telomeres also contain repeated nucleotide sequences, for example, repeats of TTA-GGG from 5' to 3' direction in most higher eukaryotes. Special DNA binding proteins may also bind to the repeat sequences in human telomeres. For example, TRF1, which helps to regulate the length of the telomere, and TRF2, which maintains the single strand extension. The telomeres ensure that the DNA molecule is replicated efficiently, and they also protect the end of the chromosome from being recognized by the cell as a broken DNA molecule in need of repair. The third important feature of eukaryotic chromosomes is the presence of many origins of replication, which ensures rapid DNA replication. Let us now take up the organellar chromosomes. First, let us see plastid chromosome. All plant cells contain one or more type of plastids and they replicate by division. A typical leaf cell contains 10 to 20 chloroplasts and each chloroplast contains about 5 to 10 copies of the plastid genome. The plastid genome is a circular double-stranded DNA molecule of about 120 to 270 kilobase in size. The plastid genome of higher plants contain three main regions, that is, a large single copy region, a small single copy region, and inverted repeats. Genes transcribed on the forward strand are on the outer circle, while those genes which are transcribed on the reverse strand are on the inner circle. The plastid genome contains some genes for photosynthetic reactions and also genes for ribosomes similar to prokaryotes. The next organellar genome is the mitochondrial genome. Eukaryotic cells contain many mitochondria per cells and each mitochondrion contains multiple copies of mitochondrial genome. The mitochondrial genome exists as a series of subgenomic circular double-stranded DNA molecules and the size ranges from 200 to 2500 kilobase pairs. The majority of proteins in the mitochondria are nuclear encoded and the mitochondrial genome contains less than 100 genes. Let us now see the conclusion. The genomic DNA in both bacteria and eukaryotes are highly compacted to fit within the small cells or nuclei. The packaging of bacterial DNA with polyamines and other low molecular weight basic proteins forms the highly supercoiled nucleoid in bacterial cells. They replicate from a single origin of replication. And in eukaryotic cells, the linear nuclear DNA molecules associate with histone proteins to form nucleosomes, which further forms a highly condensed structure called chromatin. Each eukaryotic chromosome contains a single linear DNA molecule and a metaphase chromosome contains two highly condensed chromatin threads called chromatids joined at the centromere. Thank you.